Hi, welcome to the ISEB CE 13 Plus Science Biology Lesson 2, Comparing Plant and Animal Cells. Let's get to the marks. Okay, all living things are made of tiny biological units called cells. Here we have an eagle, a plant, a fox, and some tiny microbes called bacteria, and all of these contain cells. The first cell that you have to know about is known as the animal cell. Can you label the animal cell and state the function of each structure? What's this structure called in the middle? This is the nucleus. This contains DNA, otherwise called deoxyribonucleic acid, the code of all living things. The cell membrane controls the entry and exit of substances. The cytoplasm is a clear jelly where chemical reactions happen. The mitochondria is where aerobic respiration happens, and this is where energy is released. Energy is what all living organisms need to do work. And finally, we have the smallest structure in cells. This is the ribosome, where proteins are made. Proteins are structural chemicals found within living things. The next cell we need to know about is plant cells. Now, plant cells have all the same organelles or structures as animal cells. They contain a nucleus, mitochondria, ribosomes, cytoplasm, and the cell membrane. But plants also have 3 extra structures or organelles. So let's look at these. The first one is the chloroplast, which contains a green chemical called chlorophyll, which absorbs sunlight for photosynthesis. The next structure is known as the cell wall, and this structure is for protecting the cell and stopping it from bursting when it absorbs water. The third and final extra structure that plants have is known as the vacuole. This structure contains cell sap, a sugary solution that contains minerals and other nutrients. Now, for your 13 plus exam, you will need to know how to describe the differences between plant and animal cells, so make sure you learn them. Animal and plant cells contain a nucleus, and we call these type of cells eukaryotic or eukaryotes. If cells are so small, how can we even see them? Well, we can't with our eye. We need a special piece of equipment that you find in a science laboratory. Some of you may have even used one. What do we call this piece of equipment? This is a light microscope. Light microscopes are really useful for looking at cells. We can see the nucleus and the cell wall. However, we can't see the really small structures like ribosomes. These are too small to be seen with a light microscope. Now let's look at how we use a light microscope. The first thing you need to do to use a light microscope is prepare a glass slide. Write down what you think is happening in the pictures below. You may need to pause the video for this. Let's go through them. Place a drop of water on a slide. Float a thin section of a specimen or sample on the water. Next, add a stain, such as iodine solution. These will allow you to see details more clearly. And finally, take a small piece of glass that we call the cover slip and press down gently to remove air bubbles. Now let's find out how we use a light microscope to look at our slide. First off, we need to be able to label the light microscope. And at the start we have here the eyepiece lens. This is what you look down. Then the coarse knob, which will move the stage up and down, as you can see in this animation. You'll also see the light source on the animation. The light source shines through the cells and enables us to see the structures. At the top here we have the objective lens. This is what we can rotate in order to select the magnification that we would like, times 50, times 100, or times 1000. Next we have the stage. This can be moved up or down by turning the coarse knob, and therefore bringing the image into focus. And finally, we have the fine focus. Once we can see the image, we can turn this to make the image clear, so we can see details. Let's look at an extension question. Let's see if we can calculate magnification. Here is an onion cell. The image A here is 35 millimeters in length. The actual size of the cell before magnification is 0.2 millimeters. So how many times has it been magnified? Well, we can use an equation. Magnification equals image size over the actual size. So the image size is 35 and the actual size is 0.2. And what does that equal? 175 times. The magnification is 175. Now pause the video and try answering these questions. Okay, let's check your answers. How did you do? Remember, you can rewatch the video as many times as you need until you know the content off by heart. Like and subscribe. The channel's Let's Get to the Marks, and good luck with those entrance exams.